Okay, so first of all, I would like to thank you very much for inviting me for this wonderful webinar related to saliva. And as you know, salivary diagnostics and salivary research constitutes like a great majority of my past research experience throughout the past 10 years. So today I would like just to reveal a little bit what our team at the UCLA is working on. So, as you know, in the recent times, we observe a great advancement in the technologies in the next generation sequencing. And those are those all high throughput sequencing technologies that are used to determine the order of the nucleotides in the entire genomes or in the targeted regions of DNA and RNA. And what is very interesting, that for example, in case of the bulk RNA, the cost recently went very much down. And we observe, for example, that that the sequencing technologies are not so expensive compared, for example, to the single cell RNA sequencing now. And actually, NIH invested a lot of funds and research investment into the development of the extracellular RNA communication. And actually, NIH established specific consortium that is a group of the people who are working on the establishing foundational knowledge and technologies for extracellular RNA research. And what is interesting, you will find a lot of publications that are related to the xRNA research in different biofluids, like in serum, in plasma, in urine, and so on. But there is just a little bit of those publications related to the salivary xRNA. And only most recently, we see a great increase in the studies related to the salivary xRNA. So what is what could be the reason why in the past there is so much studies being done on the saliva and salivary xRNA? So maybe I will be able to give you the answer, the quick one. So actually saliva is very, very unique. And as you know, the amount of the RNA within the saliva is very, very small. So many teams were trying to extract enough RNA amount to be able to sequence those data. However, there was not everyone succeeded. So actually I was very lucky I joined as a postdoctoral fellow, the salivary diagnostic laboratory at UCLA under the supervision of the Professor David Wong. And our team was working on developing the best methods for the isolation of the RNA from saliva, as well as for the library construction for the next generation sequencing studies. And we published this in the Clinical Chemistry. And what is unique, that actually our team was, when I joined the UCLA, we're working on the clinical utilities of salivary extracellular RNA. And what is very much important, that our study was funded by the NIH for the 5 years, and NIH invested $5 million in doing it. We used gastric cancer as a clinical example to study actually the salivary extracellular RNA utility. And we performed a big project, because why we use the gastric cancer? Our lab actually developed a very fruitful collaboration with the Korea. And in Korea, you know, gastric cancer is so prevalent. So we could get a lot of, like, enormous amount of the clinical samples for gastric cancer. So we performed the biomarker development according to the probe design. So we did the prospective procurement of the saliva samples. We did the biomarker discovery on 100 gastric cancer samples and 100 control samples. We did a small verification of the biomarker candidates and definitive validation, two phases of the validation. So the first one, we checked the performance of those biomarker candidates in 500 gastric cancer and 500 control samples, and we developed the panel of 10 best biomarkers. And this panel was validated in the second step in 250 gastric cancer and 250 control samples. And currently, we are advancing this panel of those salivary xRNA biomarkers for the FDA approval. So, as I mentioned, we use gastric cancer just as the clinical example to explore more the role of the salivary xRNA. But what is unique about the gastric cancer, it's not so prevalent in United States, but the numbers are a little bit increasing now. And what is important specifically, that when it's diagnosed very late, at the stage 3 or 4. So it doesn't give any early symptoms. And in Korea, it's very much prevalent. Maybe it's associated with some specific risk factors there, like, for example, the viral infection of the Helicobacter pylori or Epstein-Barr virus, maybe due to the spicy diet that they are doing, a lot of smoking, alcohol, and so on. But in Korea, they have a screening program. So they are performing endoscopy every 2 years for each of the people over 40 years old. But in United States, we don't have any screening tool for detection of the gastric cancer. And if we could get, for example, this salivary xRNA test to check for the gastric cancer, we know that we could save a lot of human lives and also increase their survival. So what is exactly the reason that there was not so much studies done on the salivary RNA sequencing? Actually, we discovered that there is no standards for the bioinformatic processing of salivary RNA sequencing data. And maybe this is the reason that undermines saliva diagnostic potential. And saliva is very much unique compared to other biofluids like urine, like bile, like cerebrospinal fluid, because it contains a lot of bacterial xRNAs, more than 60%. And when NIH was developing the quality control for all the biofluids, they discovered that only saliva was not passing their quality controls. All the other biofluids, serum, urine, they did, but not the saliva. So actually we're working with them to develop specific quality controls that can be applicable to all the biofluids, including saliva. And the same, they developed their bioinformatic pipeline for analysis of the RNA sequencing data. But only saliva could not be processed through it. So we work with them to adjust this pipeline specifically also that saliva could be processed through it. And we concentrated about developing and comparing many approaches for the bioinformatic analysis of those salivary RNA sequencing data. So we can get the standards for the human saliva. Oh, someone was drawing on my screen. Okay, so we developed many pipelines. We compared them, and we chose the best one that we published in our paper in the bioinformatics in 2017. And what would be just computational processing of those salivary xRNAs through all those bioinformatic pipelines if we don't have the clinical utility? So for us, it was very much important that the panel that we discovered for the salivary xRNA for gastric cancer, that it would be validated in a new cohort of the clinical samples for gastric cancer. Therefore, we took this panel of the biomarkers and we validated in a new cohort. And this is our most recent published papers in cancer. So the conclusion, my takeaway message for you would be that if you are going to work with the salivary xRNA and process your data, pay attention to the high content of the bacterial reads and the bioinformatic analysis. Because of this high bacterial content would require some specific steps. And if you are going to work on those, please feel free always to reach out to us, so we will help you to process those data and maybe to improve even our guidelines for processing those human salivary RNA sequencing data, specifically for the biomarker development for many diseases. Thank you very much.
We'll just go ahead and have our second presenter, Dr. Christopher Hiney.
Great. Thanks so much for the invitation to be here. It's so wonderful to see everybody virtually online. I'll be talking today about some of the serological applications of measuring antibodies in saliva and going through a few challenges and opportunities in this space. So hopefully we'll be able to let you recognize these challenges and opportunities and figure out that not all saliva is the same. We'll give some examples about the types of infectious diseases that might be well-suited or not well-suited and their potential implications for clinical and just general public health decision-making. And then some examples of how some multiplexing technology and combinations, conditions in sequence and kind of algorithms can optimize some of the performance of these salivary pathogen-specific antibody assays. Okay, this is my group. I'm at Johns Hopkins in the Bloomberg School of Public Health. We have a salivary biomarkers of infectious diseases program that largely got really kicked off and got a lot of momentum as a result of the pandemic response. And we have a lot of big team, collaborative teams to thank for all of our advancements in this space. So one person who was at Hopkins when I started here in 2011 was Doug Granger. He had an interdisciplinary institute for salivary biosciences. He's now at UC Irvine. He's a real thought leader in the salivary biosciences space, and he had always been teaching me a ton about all the diverse applications of saliva to different fields. And I was coming at it from infectious diseases and pathogens. And so we had contributed a chapter to this book, which is just sort of a reference point. It's a nice reference. It's a nice text on all the broad applications of saliva. And what I'll focus on from now on is our contributions in pathogen exposure. So prior to the pandemic, we were really excited about certain things. I'm in an environmental health and engineering department and thinking about climate variability and climate change. We were really excited about things like arboviruses, where vector-driven expansions can be occurring. There was a Zika outbreak that made its way into the southern portion of the United States. And we were starting to think about, for some of these infectious diseases, could we build out a tool that's non-invasive and can get at population scale for looking at prior natural history, part of exposure, attack rates, without people having to go in to a clinic and get a blood draw. And you could do this sort of through saliva at scale. And so we were seeing really nice results with dengue. In saliva on the left, you see the kinetic curves, the order of the different virus-like particles for each of the dengue serotypes. They match in saliva on the left with the serum from the paired time points in people. And so this is really exciting and really good. But then the pandemic was upon us, and we said, okay, let's see if our experience in saliva could be applied to the pandemic, and really specifically go into the areas of saliva as a surrogate for a blood draw where you're looking for antibodies, typically in blood, as a reference method. And so there's some caveats and there's some background in that not all saliva is created equal. And what do people mean when they say they're testing saliva for antibody responses to any of these pathogens, particularly SARS-CoV-2? And so as a surrogate for your systemic humoral immunity, there's certain things that are good. IgG is really good. IgM is not that great of a target. It's a bigger antibody. It's a bigger molecule. It doesn't make its way in the natural leakage from the blood into the transitate into the mouth. And so all of these different kinds of saliva, whole saliva, parotid fluid, sublingual, submandibular, gingival clavicular, they can give you different insights into the connection between your mouth and what's going on in your mouth. And maybe if you're interested in systemic humoral immunity in blood, then maybe you would be looking for some things at the top here, gingival clavicular fluid or oral mucosal transitate. And if you're looking in the blood, in the mouth, and you want to get something closer to mucosal immunity, you're down to the bottom, the cooler, the blue regions, and maybe whole saliva would be okay, parotid fluid or sublingual or submandibular fluid. And so kind of as a reference, somebody who's done a lot of work in this in the past is Per Brunsteig, who's done really seminal work on this. He's sort of the father of mucosal immunology, and has done a lot to characterize this connection between systems of our immune and antibody production. So immunity in terms of mucosal immunity is also important for the pandemic, because you could have varying immune responses and have different levels of protection depending on whether you encountered this SARS-CoV-2 naturally through an exposure, to the virus and your immune system ramps up, versus if you got a vaccine that you got through an intramuscular route, which is the vast majority, if not all the vaccines, versus if we're going to have intranasal vaccines, saliva could play a big role for all of these kind of inferences from blood into the mucosal responses. And so when we're interested in doing this, it's important to think about collection. We collect gingival crevicular fluid, which is highly enriched for IgG that comes from the blood. So things that you're measuring with a sponge in the mouth through this, which can be done very non-invasively among children. Here, you can do it, self-collected children can do it at home without a health worker. And here's another even younger. And it works until sometimes you have to have a plan for when a child reverses the swab and puts the stick in their mouth. And how do you detect and see and spot that saliva was not collected appropriately, and maybe you're not getting the right inference that you're looking for in terms of antibodies. So this is something that we've got swabs that are enriched for saliva collection for antibodies. Here, there's several companies that do this. OraSure is one that's been around with the HIV antibody diagnostic for a long time. We tend to use the Oracol because it doesn't have any buffers or additives to it. And then others like the Saliva Bio by Salametrics has come up with an oral mucosal translated device. But really, at the end of the day, the main point is there's differences in the level of antibodies that you get out of these swabs. Here on the left is the OraSure device with the buffer, and you see the total IgG levels are much lower. So the amount of that serum-driven IgG in that saliva sample is much lower when it's diluted. And then here's the Oracol that we like, is just a sponge on a stick that just harvests that gingival crevicular fluid. And then this is the Salametrics device, which doesn't quite get as uniformly high amount. And to drive this home, the beginning of the pandemic, we were thinking one sample, two tests, antibody and RNA. And all the RNA tests for SARS-CoV-2 were people spitting in tubes, spitting in cups. And so we thought, what if we did our antibody test, our swab, where we just had people spit into the tube? And so you see the diluting effect of just spitting into the tube. It increases the volume, you have more material to work with, but it dilutes that signal you're looking for in the serum-derived IgGs. Okay, so you see this big shift downward in the total IgGs. So that's really the most important thing, and applying this experience to the pandemic can give you a lot of insights and capacities, but also can create some challenges. And I'll spend time talking about some of the challenges and opportunities now. So if you're looking at, can you do serology for COVID-19 at population scale, we see really nice correlations between saliva on the y-axis and serum IgG on the x-axis. Okay, really nice correlation. So, and really good diagnostic accuracy here for whenever you're past 14 days of confirmed SARS-CoV-2 PCR positive people, that's 14 days after their symptom onset of COVID, you see really, really exquisite sensitivity and also very good specificity. And this is on par in saliva with a lot of the diagnostic accuracy that these FDA cleared serum-based tests have. That is like the serum, a serum ELISA for SARS-CoV-2 IgG, for example. And we can, through the technology that we use, we multiplex and across a range of different antigens to the nucleocapsid, which reflects natural infection, or to the receptor binding domain, which is the target of the vaccines. We see consistently high combinations, and algorithms can give us really exquisite diagnostic accuracy for the antibody responses to SARS-CoV-2. So heterogeneity is an issue you have to think about. Remember that picture I showed where the child reversed the swab and put the stick end of the saliva collection device in their mouth? How do we spot that? So we can combine that SARS-CoV-2 or any pathogen-specific IgG test on the multiplex technology we use with a test that answers how much total IgG of all pathogens, every single target, do we see in the person's saliva sample. And when we do that, and we say we need to have a minimally acceptable level of total IgG, if we're going to call a sample a negative for a pathogen-specific signal, and then we can see a real improvement in the red bar, which is the sensitivity of the assay. And without any qualifier of total IgG, you let all of those salivas that were collected, maybe in a suboptimal way, they're in your test accuracy. They're making it through the door. They're not being triaged out, and your sensitivity is really compromised. So when you start to up that requirement of having a greater quality control for total IgG, that's the gray bar, you see the sensitivity improves, but you have this class of saliva samples that you're saying you're flunking them. They're an indeterminate class. They're insufficient to be considered negative, so you exclude them. And so one of the challenges that comes along with this really nice technique is it cleans up your sensitivity. And in a testing sequence where you would just ordinarily do this multiplex assay for IgG, you say, is it positive? You would clear it. But if it's negative for SARS-CoV-2 IgG, you ask, what's the total IgG level? And is it above 15 micrograms per mil, which is our qualifier? If it is, then it goes on and we say it's a true negative. It's okay to go through. But if it's not, we disqualify and you repeat collect it. That can clean up your antibody kinetics. On the left, a messy kinetic with no qualifier of total IgG. And on the right, after you qualified, it cleans up a lot of those signals that you would say, wow, for SARS-CoV-2, they're really low. These are all people in these plots who have had a confirmed SARS-CoV-2 infection, and it can really clean things up. But it leads to a fair amount of disqualifications of saliva, which is inefficient and costly when you're talking about, study budgeting, people's time to be able to get to work on these studies and participants' time to be in these studies. So we see whenever we observe the collections, we can manage that percent of samples, saliva samples that are flunked, and keep it quite low. Even with an at-home video observing, it gets up a little bit. Whenever you don't observe this, you just let people follow, say, an instruction sheet, or you give them a link for an instructional video, we see that this percent of insufficiently collected saliva swabs can really be high. And it's of note in studies where we've done this in children, it can be really high, it can be the highest, up to 60% of saliva samples just don't pass this quality control. So that's an important thing to manage. We've teamed up through the pandemic with big collaborative scientific communities, including some modelers, and some modelers at the National Institutes for Standards and Technology, or NIST, have really come up with a modeling approach to take this total IgG information into the assay with a more complicated modeling system than I know. But they can essentially create a decision theory that really manages and reduces the indeterminate class. So they're able to reduce this indeterminate class by 40% by creating two-dimensional, and in some cases, three-dimensional modeling around diagnostic classification. So that's really, really nice and advancement too. But I think I'll just kind of wrap up by going into some of the opportunities, even with this challenge. If you're talking about a saliva test that has really good diagnostic accuracy, what can we do? Well, we can do things in populations where it's really tough to do needle sticks. And this is in children and school-based settings. We can get inference, even with insufficiency, we can get inferences about the kinds of attack rates, the natural history, that is the anti-N positivity in different age groups that are tough to reach. We can also get more granular in this and identify risk factors. And we can look at the classical risk factors about household crowding, household income, and workplace, and going to work and working in a place that has close contact with other employees as risk factors. So all this can be done with saliva. Additionally, other vulnerable, hard to reach populations, underrepresented communities of color is where we're doing work in Flint, Michigan. We've done work in Howard County with first responders, and oversampling of underrepresented communities of color to really reveal all through saliva differences in disparities in terms of the attack rate of SARS-CoV-2 with African Americans and Hispanics showing the highest percentages, and again, breaking it down by age groups, including children. And we can also, through this multiplexing assay, get at, through saliva, the effect of background on vaccination and also natural infection. So it's something else that can all be done within the context of the available vaccines. Just to say that we've taken next steps to ask how this salivary IgG response correlates with plasma neutralizing antibody titers, because it's a big thing. What's the durability of our neutralizing antibody titers? And for our saliva assay, it looks like it's quite comparative. And for certain antigens within our multiplex, like this Mount Sinai S, we see that the percent agreement is just as high between our saliva assay and neutralizing antibody as it is for the leading blood-based serological tests. These are FDA, EUA approved assays from Euroimmune, Biorad, orthovitros, which the FDA has been using to qualify convalescent plasma for donation in the convalescent plasma trials. So that's really important. And if saliva is a surrogate for neutralizing antibody, you could potentially have a tool to monitor population immunity over time. And that's something that we've done to look at durability to natural infection over time. And if we compare our durability in saliva here and the half-lives that we see of 64 days, 100 days, and 148 days really are highly comparable between saliva and the estimates from some of the leading groups in blood. These are the blood-based serological tests from people like Florian Kramer and Mount Sinai. So that's something that could be a really effective tool to monitor population immunity at a population scale, look at biologic factors like sex, symptomatic status is a predictor of relationships with the degree of how symptomatic people are. And look at natural infection versus vaccination, and also look at breakthrough infections. The last thing I'll end on is in saliva, beyond the idea of getting a surrogate for systemic immune responses to, say, blood-based IgG, we also are harvesting the antibodies that are the mucosal antibodies. These are the IgAs, the secretory or component IgAs, which are the front line of defense as we ingest things into our mouth and there are pathogens or all kinds of other stimuli. We have robust antibody responses that are these secretory IgA responses. And when we look for SARS-CoV-2, that's SIGA, and we take account of the order of either infection and or vaccination, we can start to see the robustness of the SIGA, or these mucosal antibody responses to SARS-CoV-2. And here is people who are only encountering the virus. This is natural infection. You see this nice increase in their mucosal secretory IgA response. It goes up really nicely between the naive and then increasing number of days since confirmed COVID infection onset. And then when you look at vaccination only, it's not as high. These mRNA vaccines, you see a little bit of a boost after the first dose, but it's really half of what the sort of peak level is among people who had exposure to the virus, right? And kind of a stable, but maybe a little bit of a leveling off of this mucosally elicited response, which you may not be surprising, and others are also showing. But then people who previously had encountered the virus and had natural infection and then were vaccinated, they really had achieved high levels of SIGA, particularly once they get their first dose. They get up to almost double, more than double of what natural infection only gets. So this nice sort of prime boost scenario, and it really looks like it might be boosting the secretory IgA, which is an important thing. And then finally, those individuals who were vaccinated and then they experienced a natural infection after being vaccinated, you do see their evidence, the really, really robust mucosal antibody responses here, up to the highest levels that we see out of all these panels. So that's something that also, thinking back to Per Bronsteig's work and thinking about your question, is it a surrogate of a plasma antibody ELISA test for in our case SARS-CoV-2 or any other pathogen, or is that pathogen interacting with our bodies and our immune systems in a way where the mucosal antibody response can be really important? So saliva offers and affords these opportunities to get both a surrogate of plasma, serological response, and also this mucosal antibody response. And so that's something that we're pushing into the future. And we could think for, be it COVID or be it any other infection, we could monitor the durability of these dynamics. Being natural infection, if there is a vaccine candidate available, you could look at these drivers of the durability over time and look at IgG and SIGA responses to these different antigen or pathogen specific targets. And you could really think, we also need to think critically about normalizing, which I didn't quite get into. And think about the mechanistic insights that we're looking for. Is it to get mechanistic insights about blood or some kind of a mucosal process? So you could also, with saliva, as others are going to point out, do at home. It's non-invasive. You could have people being a participant in learning about their infection and immunity and be doing it from the comfort and safety of their own home. As Anne and the Saliva Direct group have shown, you can combine RNA and antibody response measurements, and you could try to optimize what we've done in gingival curvicular fluid for passive drool, and really ultimately think of this as a core tool to measure correlates of protection over time. So thanks so much. And I really appreciate the opportunity to speak to you.
Hi, Chris. That's a really good talk. Yeah, I learned a lot with that. I'm just curious. I have a bunch of questions. I'm just curious how the IgG is. You meant the IgG are sourced from serum. What is kind of the circulation? How that originated from the in the saliva? It's like more compartmentalized.
That's a great question. So we have a consistent leakage of the blood-derived components that sort of leak into our mouth. A lot of that leakage happens at the juncture between our gums and our teeth. It also occurs in the back, in the crevices of the mouth and the juncture of the mouth, where there's just also consistent leakage into the oral compartment. So people who are adential, I get that all the time. People who don't have teeth. There's also leakage processes for the blood-derived components to leak into the mouth. We've done some studies in newborns where we can still detect IgG constituents of it that come from the blood in early phases of life, days and weeks into life. And to give you another really concrete example is that when each of us sleeps, if you're in the typical daytime awake and nighttime sleeping schedule, whenever you sleep, there's a constant continued rate of leakage let's just take IgG from the blood into the mouth. And many people suffer from sort of a dehydration or a drying of the mouth overnight. So there's this natural concentration of that consistently accumulating IgG overnight. And when you wake up is one of the times of day when you have the highest concentrations of IgG and other blood-derived components in your mouth. And then as you, I don't know, take your first sip of water, brush your teeth, take a sip of coffee, eat breakfast, and go about your day, you start to flush and wash and dilute. And also diurnally, we just kind of like have this diurnal pattern of that morning rise and down to a decline over the day. So it really is that biology of consistent leakage of those blood-derived targets into the mouth. And either by thinking about the swab collection device to really harvest those blood-enriched targets, or just by thinking about things like what time of day would you have the highest levels of these blood-derived components, can really help you a ton if your goal is to get like a good surrogate for blood-based, say a blood-based reference assay.
Thank you so much again, Dr. Heaney. We'll go on to our third presenter, Dr. Kulhi.
Thank you. Thank you guys for inviting me and showcasing what we have done over the past 2 years. My name is Ravi Kulhi. I'm a pathologist at Medical College, Georgia, Augusta, Georgia. And what I'm going to talk is a little bit more on the clinical side, how we have taken this as a sample type in both on the clinical testing as well as research testing for infectious disease. These are my relevant disclosures for the content. I'm a healthcare provider in the state of Georgia, and the opinions are personal. So outline for the next 10 minutes is I want to give you the reason why we got into this, what were the challenges, and what is the current utility and how we want to use saliva as a sample type in the future. So earlier part of the pre-pandemic, around second or third week of March, I was asked to be on the governor's task force for the state to figure out the massive testing for the entire state. One of the biggest challenges, along with my colleagues from other academic institutes, I realized was supply chain constraints. Early we were targeting at least 10, 15, 20, sometimes 30,000 samples a day. Again, you got to remember the reference is, this is the third, fourth week of March 2020, the first week of April, where pretty much everything was non-existent. And there was a huge rush to start figuring out actual collection devices, like how we're going to get samples collected. And especially in the rural and rural hospitals in the state of Georgia, pretty much everything was exhausted by the time even we started the massive screening program for the state. So the question came, can saliva be used as a sample type for COVID-19 screening testing? And then is it a true surrogate for nasopharyngeal swabs on the clinical side? I mean, I know there was a lot of research going on. So I directly reached out to FDA. FDA was very receptive of the idea, and they said, but is it, we already had an EUA in mid-March from the lab as an LDT, and we wanted to add saliva as a sample type for that particular application. So FDA said, yes, you can do it, but you have to do it, and they sent us a protocol to do it. But our problem at that time was there's only one existing protocol, which was, I think, a red dress protocol, which was mostly done on inpatient, and the indication was for clear saliva collections. Our problem was we had 30 drive-through collection sites all across the state. We were getting samples from ICU, nursing home, and a lot of people were sitting in the drive-through for an hour and a half, 2 hours to collect the samples, and we would get people who eat, drink, and they would get this pink salivas, red salivas mixed with the food. So it was a huge challenge for RNA extraction, and pretty much most of these were getting invalid during the PCR testing. And then the samples which we were getting from ICU, nursing homes were very viscous as compared to what the original protocol in April was for clear saliva collection. So this was way before Saliva Direct existed. So what we did was we partnered with Georgia Tech and trying to really figure out how we can take the salivas we were getting and make meaningful use out of that sample type. And one of the things we realized after multiple back and forth with some of the smart engineers who are sitting home in Georgia Tech was we could do a bead-based homogenization. So when we started collecting samples from a hospital setting, nursing home, communities, and drive-through, we started collecting them in these tube which had collection media as well as beads in the bottom, and we would put them on a bead-based homogenizer, and they did two purposes. One, it converted the saliva, whatever way we were getting it, into a very clear fluid, and which was very easy for extraction, and that pretty much solved most of our problem. On top of that, post bead-based homogenization, you're able to pool them based on, again, this is way before even FDA was accepting pooling as a method for screening. But we showed in our validation to the FDA that you could easily pool post bead-based homogenization 1:5 or 1:10 sample, and then increase our capacity for screening purely because we were able to do the bead-based homogenization. It converted the viscous or whatever the saliva we were getting into pretty much very clear fluid, or fluid which was helpful in getting the RNA extraction and helping us to do the PCR on that. So we were able to publish this data and very well accepted now that we could not only get a post bead-based homogenization, we would get a very nice saliva for extraction, plus also the pooling out of that. In the last 2 years, we have done multiple things, especially the supply chain constraints, and publish all that information to help us prepare for the next or even the current pandemic. And one of the papers was we went through each and every aspect of the entire testing process, right from sample collection, how to make VTMs, how to make 3D printed swabs, how to use saliva, how to make saliva kits, saliva collection kits, because suddenly saliva became such an interesting sample that we could not even get saliva collection kits. So we developed our own in-house kits and then all that. After that, again, the challenge became extraction. So yes, we were collecting saliva, but there was no RNA extraction kits at that time. So the question was, can we directly use saliva without and bypass the RNA extraction protocol? And again, on the research side, I think there were a lot of work done, but what we were trying to do was how we can take this and convert into a clinical protocol and submit for FDA inspection and EUA. So we developed this research protocol where we would take direct PCR, and then one of the terms we were using for community-based screening was spit in a cup. So you take this one cup and go to a family, and then all the 4 or 5 or members would spit in 1 cup, and that would be labeled as single sample, a pool sample, and then you could use that. If that becomes negative, then you don't need to go back and ask for any further intervention. But if that comes positive, then you ask the family to quarantine for the sake of pandemic. And this really was really helpful. We have worked with countries in Asia as well as South America. Spitting in a cup would really help them to maximize their mass cleaning protocols, especially with the countries which have a high population. So on top of that, you could pull these samples. And the next step was when the winter came and we worried about combination of flu as well as SARS-CoV-2, we tried to see if saliva can be used as a true surrogate for nasopharyngeal swab to look at SARS-CoV-2, flu A and flu B, and then we were able to show a pretty comparable validation. And again, saliva is a true surrogate sample. So next time when you go see your primary care physician, do definitely ask if you can use that instead of having the pain of going nasopharyngeal swab. So currently we have 3 clinically validated protocols in the lab. Saliva, all basically a platform or a technique which is helping us process saliva samples from all available sources. Saliva stat, which is not that much used because now we do have RNA extraction kits available. But again, this is a really really good method where in case of supply chain constraints, you could bypass the RNA extraction and pan saliva. If you want to test more than one analyte in infectious disease analyte in saliva, you're able to do it, which is comparable to nasopharyngeal swab. Again, this was during the pandemic, and we were interested in sequencing all these different targets, and most of, if not all, our samples were saliva. So the question was, would saliva be a good way of to not only test but also sequence that? And then we showed that saliva is really viable sample type for sequencing these respiratory, all the 40 different various types of viruses in that panel, 8 different viral families. And we even went further and created a molecular AP program for the state. Divided the state in 3 different parts, and basically based on the zip code, we're able to sequence all these viruses and put it in our next strain, which basically shows how these the strains were moving in the state during that particular, I think, 8 to 10 months of time. Again, the idea is to prove that saliva can be a true source of sample type for molecular AP. We also looked at microbiome, both nasal as well as oral, and we were able to, both in infectious disease settings, especially the individuals who are severely infected or symptomatic, the saliva really help us to understand the microbiome, oral microbiome in these individuals. Another application of saliva, we have a longitudinal study going on for the last 2+ years called SPARTA, which is based on SARS-CoV-2 and COVID. And then the reason I bring this up, because anytime when you have a longitudinal study which is multi-center, the compliance of sample collection, we do initially collected samples every week, and now we're collecting every month. Saliva was such a great method of collection, and the compliance was great to the extent that now we have literally thousands and thousands of samples of saliva. And in parallel, we have a pretty significant amount of parallel serology on these patients. So you can see there are only 25 subjects consented for nasopharyngeal swab. Majority of them have saliva as a sample type for this longitudinal studies in healthcare workers over the last 16 months. So it's again a great sample type, great tool, which is helping us with compliance for these kind of study. Another huge advantage for saliva as a sample type was when we were going outside health system, we went to increase our enrollment for the SPARTA study for minority and undocumented people, and that's where we went to African American churches and local Black barbershops. This was a setting, and you could clearly see saliva as a sample type played a huge role for enrollment, rather than if we were doing a nasopharyngeal swab. The compliance would not have been that much. Just to give an example, in our sites, we had around 37% of participants who are minorities or rural, versus the other side, which was around 2.5 to 3%. So it's a huge difference of compliance, and in a community-based setting. So, and in summary, saliva is a true surrogate sample type for clinical testing. It's a very easy of collection. And I didn't even talk about storage. It's a very stable at room temperature. The processing is relatively simple. Again, it's an effective sample type and constraints in supply chains. I mean, I don't want to go back in the first or second week of April. Literally, there were 0 swabs, and we were asked to process 30,000 samples a day by the state and the White House. And basically, saliva literally was a game changer in bringing in the community-based mass screening at that time. It definitely has a less exposure for collecting sample. Most, if not all, the saliva is collected by the individuals themselves with a self-collection, especially in a drive-through or community setting. That definitely helped us with our IBC and IRB approvals. And again, for longitudinal studies where you have to collect the samples every week or every 2 weeks or a month, the compliance from subjects for a clinical study is amazingly different, amazingly good. And you'll have effective studies where saliva is a sample type, self-collection is the method of collection. And if you use a method like a bead-based homogenization to extract the better value of the saliva, I'm not sure how much of that would affect Chris's work with IgA and IgG. We have not looked into that, but at least for the RNA and PCR-based testing, the bead-based homogenization was extremely effective. This is my lab and an amazing group of people who have worked over the last 2.5 years to make all these things possible. Amazing group of people. I mean, we pretty much were supporting the entire states of COVID testing for 30, 90 days. And I'll take any questions.
We'll just move forward with our 4th and final presenter, Dr. Tall from MIT.
So I am really excited to speak with you all today, and I could not have had a better introduction for what I'm going to talk about than what Chris has shown you. You know, I think Chris did a really great job of getting this concept across, that we have very different antibody responses that are important for protecting our, inner organs and our, what we see circulating throughout our blood, and what we think of as our systemic immune response. And what we have, the antibodies that protect our mucosal barriers and essentially keep these infections from coming in. And saliva is this really interesting sample type because we really get to have a view of both worlds. So we get a little bit of that blood, that's the blood components that are leaking into the saliva, but we also have this really nice window into these mucosal antibody responses. And these are really important for indicating to us about protection from infection, not just protection from disease. And of course, during this pandemic, this became a really important topic because, as our first vaccines started to roll out, these were vaccines that were very focused on protection from disease, hospitalization, and death. And now as the conversation really shifts towards vaccine types that could really provide protection from even getting infected in the first place and being able to transmit, and infect others, we really need to look at different antibody types that can mediate this protection and their location. And so location becomes a really important factor, and saliva becomes this incredibly valuable sample type where we can really look at both, but we have this really great way of sampling those antibodies that are at the site, that are going to be at the site of infection. And this was such a huge question because, you know, this really comes down to, do we need masks? Do we not need masks? Are we protected from infection, transmission? And in trying to, you know, ask these questions, and also, you know, especially earlier in the pandemic, people were really looking to see, you know, can antibody tests serve as immunity passports? Can we really predict who is susceptible to infection and who is protected? And so in looking at that, what I want to do is to discuss two things about what we can learn from these saliva samples, both about our response to infection and our response to vaccination. And so what you've been hearing in some of these earlier presentations is that we can use these saliva samples and we can actually directly detect the infection, but we can also detect antibody responses to the infection. And you know, most of these antibody responses are focused on these, you know, general types of antibodies, IgA, obviously, which is, you know, a very important antibody type in the saliva, but also IgG, which is very important in the blood and can be detected in saliva, and also IgM. But the thing is that all of these types of antibodies actually have many subtypes to them. And it's slightly more complicated than we often think about. And so one of the first things that we did when we started our saliva testing is that we compared different saliva collection devices. And this is actually very similar, I think, also to you know, what Chris was showing, but there's some really big differences in the antibody responses that we can pick up from either passive drool, where essentially you just spit into a tube, compared to swabs such as OraSure with a preservative, or we also tested swabs including supercell and puricell, and then I'm going to show you also some data from Salvet, where those are all essentially dry cotton swabs that don't get submersed into a preservative. And what I'm showing you here is that as we look at different antibody types and subtypes, we see some really big differences between these devices. And you can see that indeed, we really can pick up a lot of these different IgG types using these different saliva collection methods. But what became really important in terms of looking at correlates of protection is that we were realizing that just the amount of antibody that you have against infection wasn't as good of a predictor of your, you know, of the extent of protection as neutralization assays, where we essentially take virus or pseudovirus along with susceptible cells and ask how many of the cells can get infected and how much of your sample can we dilute down and still have, you know, protection for these cells from getting infected. So I'm going to show you how we actually adapted this assay, which is shown here for blood, and shown that, you know, it was possible to take people's blood and really get very good indication of how protected they were from disease after vaccination using these neutralization assays. And we converted this for using saliva instead of blood in order to predict protection from infection at the site of infection. And so what I'm showing you here at the bottom is how we do these assays, where what we did is we took a VSV virus and replaced its outer glycoprotein with a SARS-CoV-2 spike protein. And we also had it fluorescing in, you know, with a fluorescent protein that we could see, and put it together with susceptible cells and then ask how many cells get infected and how much saliva do we need from a particular person in order to protect from infection. and this could give us a really good indication. And so I'm showing you what that looks like as we actually look at it. So what you see is that this fluorescent protein is green, but using computer software, we can trace all green cells with purple so that we have this nice mask where we can very easily see, based on how many cells, how many of them have turned purple. I'm going to show you this here where you're seeing all the cells are in yellow, and you're watching as we add on virus. And I'm first going to show you what this looks like if you run this assay without neutralizing antibodies. You see that you are seeing a lot of these cells turn purple, and then more of them turn purple. And you can watch this over days. And we found that this 3-day time point that you're seeing now is a really great time point to compare across everybody in terms of, how much protection they have from infection. So on the left, I'm going to show you where we're adding in neutralizing antibodies. And it's the same virus, same cells, but here there's protective antibodies. And you can see that this makes a very big difference in terms of how many of these susceptible cells or infectable cells can get infected. Now, I showed you that we tested different saliva collection devices, but the really important thing is that some of those, the collection devices that come in preservative, as well as with passive Joule, we had a really big problem that adding in that saliva at the level that we needed to in order to measure protection was killing the cells that we were trying to test. And so here, what I'm showing you is when we ran this assay with these salivette devices, this is essentially a cotton swab where we then spin out the saliva that's been absorbed into the swab. And we could compare across different vaccination doses and strategies and time points and see really big differences in terms of how much saliva was still protective against these susceptible cells getting infected when exposed to this virus with spike protein. And this pulled out really, really interesting differences for us. We have a study that specifically focused on what was going on here with the J&J elicited responses. And here you'll notice that that actually looks very different from what we saw with other adenoviral vector vaccines, such as AstraZeneca, which is on the left. So on the left in blue, you have AstraZeneca, and with or without following with mRNA vaccination. Then you have J&J with or without following with mRNA vaccination. And then you have in the browns, red, orange, you have mRNA vaccination, 1 dose, 2 doses, and then 3 to 6 months post 2 doses. And where you really want to see these samples to see any kind of protection from infection is you want to see them way down at the bottom where they can really prevent the infection of these susceptible cells. So where there's a lot of neutralization activity and you don't really want to see the samples up at the top. What we were seeing, as you can see from the brown to red to yellow, is that, as became later very clear with the mRNA vaccines, we had protective responses, which were very transient. And so, we are seeing protection that's there for several weeks, a couple of months, but as you get to these, 3 to 6 month marks, you're really seeing that a lot of people are losing that protective components of antibodies that they had in their saliva at earlier time points post-vaccination. And so if people are interested to read up more about this, we actually have a review that came out very recently in Immunological Reviews, where we really talked a lot about the different research that was done using saliva in order to observe different antibody responses, and our contribution of the using saliva to look at neutralizing responses and how those either compare or contrast to other indications that can be seen just by measuring the antibodies alone. We worked very closely with Jennifer Gomerman's group at University of Toronto And one additional thing that I wanted to mention is that my group has moved to MIT, and we have just launched this really exciting study that we call MIT Meister Study, and we are continuing to use saliva as a sample type that we're investigating as we start to really probe much more deeply into infections that really trigger chronic diseases that we don't understand. And so here we're starting our focus with Lyme disease and with long COVID. And saliva is just one of the sample types that we're looking at. But as you heard, a really great sample type, especially for longitudinal samples, where you can get a lot of information from people over time. And I want to thank the funding that we had to do this research, as well as all of the people who helped us with our studies at our saliva study, COVID-19 saliva study at Stanford, as well as continuing to work with us now at MIT. And up in the upper left, you can see our whole group of people who are working with us within the Weissman Lab, focused on saliva study, and as well as on the bottom right, we have a lot of people who we're working with very closely now at MIT and the Griffith and Laufenberger labs. And I want to especially mention some of the key people working on saliva study from Stanford, and you see Erin Sanders here and Georgina Haas, who are both still continuing to work with us at MIT. And I would love to take any questions.
Thank you, Dr. Tall. So yeah, the floor is open for questions.
This is Kirtu. My group has early on detected Lyme bacterium DNA on our BNS, which is a new type of a sensing platform. I wonder, is Lyme also detectable from saliva?
Go ahead, Dr. Heaney.
Hi, I really enjoyed that talk and that presentation of the ability to get neutralizing activity out of saliva. Could you just share how complex or how practical, or what is the complexity of that assay? It was great to see the saliva collection. Seemed like it was pretty. It was sounded like you were leaning towards the more simple without buffers. But what about when you get the saliva back to the assay? Does it take really specialized equipment, or is it something that could be that others could be trained in using and thinking, as this is the next frontier of like how to safely stimulate mucosal responses to things like SARS-CoV-2? Could there be other, how easy would it be for others to be trained or to learn the assay?
I think in labs that are running at BSL-2 or BSL-2 plus safety, I think very easy, and happy to discuss in more length. I think the thing I want to really make sure to mention is that when we tested OraSure samples, and we have a supplementary figure on this in our paper, but when we tested OraSure in our viral neutralization assays, we saw that the high salt concentration of the buffer was really toxic for the cells. And so we had to dilute it down to 1 in 30 in order to not have toxicity on our cells. But 1 in 30 really escapes that level of protection that we're seeing in the saliva. So it's just like with antibodies, we saw our best dilutions to see good antibody responses were really either anywhere from undiluted to 1 in 20 was about as low as we could go to really reliably look at the antibodies there. And the same is really true for getting a really robust read on the viral neutralization assays. And so only being able to look at under 1 in 30 dilution, you really just get the top people with, the most robust protection, though there would have been protection that you would have been missing. And so with the Salivet, what's really nice is that we actually really like to look at that. The figure I showed you across all those different vaccination strategies, that was looking at a 1 in 12 dilution, which we really like. Anything in the kind of 1 in 10, 1 in 12 are really great dilutions for us to see broadly and to just broadly compare. I'm not, I think the next step that would be incredibly meaningful is to do these longer studies where you follow breakthroughs and ask how much is important for protection, because for blood, we know this. For blood, we were able to understand that neutralization assays really needed to be at a dilution of at least 1 in 400 to mean anything about protection from disease, and ideally 1 in 800 or even able to dilute lower. And so an important question for saliva is most people at a undiluted or 1 in 6, most people have protection. And so we focus more on that 1 in 12 because that's where we're seeing really nice range across the samples and what was fitting with what we were seeing in general coming out of the patterns of these vaccinations. So we think 1 in 12 is a really important dilution. But, maybe it's important who can get down to 1 in 30, 1 in 60 and still have protection. So, but this is, the way that we ran this off of a VSV. And so, we did it using an IncuSite so that we could really run this high throughput and have software that we could get good analyses. But I'm happy to discuss this in more depth and share protocols. But I do think it's feasible.
One follow-up quickly is that were you at all concerned with the dirtiness of saliva compared to, say, serum or plasma in terms of the bactericidal potential? A lot of people talked about saliva as being so icky and creating problems with cell lines or just the bactericidal killing of cell culture lines.
And that was a big problem that we had with passive drool. And that's something that the cotton swab really cleaned up for us. I think one of the problems we had with passive drool is just the huge range in viscosity of different people's saliva. And then that had a really big impact on the assays. Using the cotton swab and then just eluting from the cotton swab, the fluid that can come out of that and using that was very helpful.
Looks like we also have Dr. Kulhi. Go ahead.
Yes, maybe, both for you as well as Chris, my question is more of what we do. So most, if not all, of saliva is directly collected, but we use bead-based homogenization because PCR was our primary focus. And do you guys think if the homogenization might disrupt some of the IgGs in saliva? Do you have any thoughts on that?
Yeah, so specifically IgG is your hardiest antibody isotype. What we saw when we tested different either heat inactivation, freeze thaws, different Triton X or other things that were added into saliva that multiple different groups had collected or were using in order to work with the saliva safely. We really saw the IgGs holding on much better than many of the other isotypes. We saw that we were very quickly losing IgE, seemed to be one of the most fragile isotypes that we tested. But IgA was also significantly more fragile than IgG. And for us specifically in our viral neutralization assays, as was shown by some other groups, it was shown that secretory IgA, the dimerized IgA, is about 15 times more efficacious in a viral neutralization assay than just monomeric IgG. And so losing the IgAs and keeping the IgGs was really, changing the sample significantly. And so I think it's something, that could be done. And if there's a lot of bank sample and it was all treated a certain way, it might be worth seeing, how it can compare back and what can still be learned about it in terms of the antibodies that are there. I do think the IgG is likely salvageable, but some of the other isotypes might not be.
Thank you.
All right, we have another question from Annie Peng.
Hi, Michael. It's great talk. What is your insight on the surrogate virus neutralization test? Like, you know, when you put like if I mix the HRP labeled RBD with saliva, and then just to detect the percentage of the inhibition of its binding to the ACE2?
Yeah, so I think that there were a lot of different assays that came out there in order to try to do this work in BSL-2 settings. And so, you know, I think we all appreciated that this is, you know, that getting access to BSL-3 for doing all the testing that we wanted to do on saliva wouldn't be feasible. And that was very important to make proxy assays that could be done in BSL-2 laboratory settings. We really liked the assay that we were using where the VSV glycoprotein was switched out with a spike protein, and that was then developed further and further to swap out different spike proteins from the different variants. However, that assay is very limited in that it's really just looking at the ability of, you know, SARS-CoV-2 spike to bind to ACE2 on cells and then allow viral entry. And we know that there were, you know, that the variants kind of differed in whether or not they were, you know, to what extent they were using viral proteases and, you know, endolysosomal entry versus just adhesion through spike ACE2. And so I think that one thing that becomes really complicated as you start to use different proxies is to how much of the native interaction you can recapitulate. So I do think there's some value to these assays where you essentially have a spike ACE2 blocking assay, and you're just trying to ask, can I block the interaction between spike and ACE2? I think that, you know, as the different neutralization assays did correlate very well, and this particular assay scored very highly on its correlation to protection from disease, which is one reason that we really liked it in terms of looking at protection from infection. But, you know, some of the pseudovirus assays didn't score as well and weren't as reflective of the biology. And so I think, you know, that's where it gets really tricky to pick what's a good proxy assay to look at if you don't want to do this in BSL-3.
Thank you.
Did you have another question? Because there was one in the chat. I think this question was directed to Dr. Chris Heaney. I believe he did answer it, but I'm going to go ahead and share it again because the audience may benefit from it as well. So Dr. Heaney, I was wondering if you also evaluated saliva collected by just spit in detection of antibodies compared to other saliva collection devices, for example, OraSure and Oracle?
Yes, we have. And the key to different sample types, similar to what Michelle mentioned, is that the idea is both the degree to which there can be variability in the swab or the sample collection types recovery of the antibody target of interest, but it's also the matrix effects itself, like what other things come along with that sample type. And so a lot of that can be managed by also adding either a second test of the same saliva aliquot or managing the aliquots very carefully when you get into really heterogeneity of a viscous or a saliva sample type. And that second test that you do is producing a signal that could represent the degree to which there is a matrix effect, or either a really tiny signal, as we heard, that you're chasing a very diluted signal, and then that being a reproducible and highly accurate second signal by which you could normalize or standardize the signal of interest, say SARS-CoV-2 antibody. We've been talking a lot about that. So when we get into passive drool, Besides our gingival curvicular swabs, which the salivette's also another sort of way to, with very minimal additives, get that at that sort of goal of the serum transitate. We really want to think about normalizing signals that we could apply. And so we've tested passive drools, and also to extend it even further into the nasopharynx, as you're saying, we've tested some nasopharyngeal swabs, which there's millions of these, right, early on, and then now anterior nary swabs stored in the viral transport media or the universal transport media. And so we see that it's doable and you can get it, but the signal is lower there. So both the pathogen specific signal, the magnitude's lower, and the total, whenever you get that total IgG quantified microgram per mil total IgG or total IgA, microgram per mil, the absolute concentrations are lower, because typically those viral transport media, you would dilute them in 1 ml, 2 ml, sometimes 5 mLs of transport media. And each time you dilute it more, you're diluting out that signal that you're looking for. So we've found ways to divide by or subtract that total IgG. And another example of just matrix effects is that if you're multiplexing, you can add a set of beads that are candidate signals that they would generate that would reflect the matrix effects, the background noise or signal. And so as you have saliva, unlike serum, where you've got all of this wide dynamic range of like background debris, junk, protein stuff, food, versus not, we found that adding a normalizing bead for that sort of matrix effect, like a bovine serum albumin bead in our Luminex assays, provides a really nice control of that, both dilution effect and the matrix effect that could be there. So anyway, hopefully that helps. And then you can also get really with that adjusting for those factors, you can get similar diagnostic accuracy across these, gingival curvicular fluid to passive drool to, say, anterior nares viral transport media is kind of the idea.
One last, if you were to choose your favorite saliva collection device, which one would that be?
If I can chime in, I would say it kind of depends on what you want to do and how many different things you want to do with it downstream. Because I think, as you heard from Dr. Coley, you really, if your downstream focus is PCR, you want to worry about the stability of the, genomic material. If you're, for us, it really depends. We're moving forward with both OraSure and Salivettes because OraSure gives us really nice flexibility on timing. Doesn't have to be processed for 21 days. It can be just kept at room temperature. The antibodies are really nicely preserved. So that's wonderful and very easy, but then it's not great for these viral neutralization assays. So for that, it's worth it for us to do Salivette, even though we have to process it same day and get it right into the freezer.
So I can also add a couple of things. I mean, it depends on 3 things. One, what is the reason you're collecting saliva? Second, the cost. I mean, when we initially started in April, when saliva became suddenly the sample of choice for most of the testing, you won't believe the amount of money they were asking for a collection device, which range from $12 to $15. And as a state, we were trying to have the actual test less than $5. Forget about collection. So we were really going for the cost, and we ended up creating something very unique. And you could split in one of those condiment containers, like the ketchup or whatever you get at the fast food, and you'd extract it, put it in the tube, and our cost was $1.30. So that's one thing. And the most important thing, especially if you're looking at as a response to pandemic, the availability of these collection kits. I mean, when we were asking for, let's say, if we want to buy 10,000, 20,000, they basically said wait for 6 to 8 months. So you really have to reverse engineer based on what exactly is the reason you're collecting. If you're looking for serology, I think you have your answer. If you're looking for PCR, you really want to go for a more stable because we're getting samples from rural parts of Georgia, rural parts of the country, transport, and then if PCR is your ultimate test, then something decide based on what's your goal and cost is.
Okay. Dr. Riley, go ahead.
And I know that we're at time, but I just wanted to speak up and just express my gratitude for all the speakers joining today, and that you've all been such an inspiration over the last couple of years. And it's been wonderful to speak with you and bounce ideas off you at various times, and just, yeah, really appreciative of, you know, everything that you've been doing.
Yes.
Well, ditto. Right back at you. And great, thanks for including us in this event.
Thank you.
Yeah, thank you.
Thank you so much to our speakers. Very much appreciate your time. Thank you.